Hello dear students. In this class, as we can see that we are going to discuss digestion and absorption of lipids. Now this topic we'll discuss in very brief, we'll not go into too much of detail, but this as a prerequisite knowledge of the lipid metabolism, some amount of knowledge you should have, so that little bit we'll try to give. So at first let us discuss the digestion and then we'll come to the absorption. So the main thing from our diet, the main form of lipid that we get is the triacyl glycerol from different types of oils and other fats. The most common source of lipid that we get in our diet are triacyl glycerol, and that triacyl glycerol or TAG or TG or triglyceride as we say, so whatever name we call it. So to break it into glycerol and fatty acids, the group of enzymes that act is known as lipase. So in digestive system, there are two main types of lipases. One is the lingual and gastric lipase, which has a minor role, and then there will be pancreatic lipase, which is the main thing. So lingual and gastric lipase, mainly gastric lipase, if you can say, obviously acts at the acidic pH or pH at about 2 to 6, 4, 5. That is usually the pH at which it acts best. Here, this lipase, it does not break all the three fatty acid glycerol ester bonds. So in glycerol, we know there are three hydroxyl groups and three fatty acids are esterified. But all of those bonds are not broken by the gastric lipase. Only at position 3 in glycerol, that we call SN3 position, only that one is broken. So here, the triacyl glycerol is broken down to release one fatty acid, and then the diacyl glycerol remains. Okay, so if we see the diagram, we can see that here, as we can see, that at position 1 and position 2, these two bonds are not broken. Only at position 3, this bond is broken. Okay, so only this one is broken. Okay, this bond is broken, not all of them. So that is how this thing acts, and obviously the product will be a diacyl glycerol and then a fatty acid. Now coming to the pancreatic lipase, which is the main enzyme that breaks down TG, helps in the digestion of fat. It obviously comes from pancreas, acts in the small intestine and acts at alkaline pH, obviously. Here it acts on two ester bonds, that is at position 1 and position 3, and it leaves the position 2. That one is not broken. So, and it is also activated by another molecule known as colipase. So this pancreatic lipase is further activated by colipase. Now here, so if TG is broken, so it will produce two molecules of fatty acids and one monoacyl glycerol, where the acyl group is attached at position 2. So we call it 2 monoacyl glycerol. That does not mean two molecules, it means the position, and then two molecules of fatty acids. So if you can see here, we can see this is the glycerol. Okay, so we can see this one is the glycerol, this one is the glycerol. So here at position 1 there is, this is at position 1 there is one fatty acid, at position 2 there is another fatty acid, at position 3 there is another fatty acid. So the pancreatic lipase step by step removes the fatty acids from this one, this position 1 and then position 3. Position 1 and position 3, these two are removed, but at position 2 this one remains. So this one remains. So our products now, so what are our products then? The products then become, this one is 2 monoacyl glycerol. This is 2 monoacyl. The acyl group is present at the position 2 and 2 amino acids are released. 2 molecules of amino acids are released. Fatty acids are released. 2 fatty acids, 2 molecules of fatty acids are released, and 1 molecule of 2 monoacyl glycerol is produced. So this is how this enzyme acts. Okay, So carbon 1, 2, 3. Carbon 1, this one released. Carbon 3, this one released. So at position 2, this one remains. Okay. Now, what happens with this 2-monoacyl glycerol? Majority of it, about 75%, remains as monoacyl glycerol, and we'll see it is absorbed like that in that form. But about 25% of the monoacyl glycerol, they are then further hydrolyzed to produce glycerol and fatty acid. That mostly is done by pancreatic esterase, but there are other ways to do it also, like isomerizing this by removing or changing the position from 2 monoacyl glycerol to 1 or 3 monoacyl glycerol. Then again pancreatic lipase can act on it. But mostly the pancreatic esterase enzyme acts on this. Then another thing other than triacylglycerol that can be present in our diet, another lipid can be phospholipids. So they are broken down by phospholipase enzymes like phospholipase A1, A2, etc. And then they produce lysophospholipid and fatty acids. And obviously cholesterol mostly in the storage form, that is cholesterol ester, that can be present in our diet where cholesterol is joined with a fatty acid. So that will be acted upon by cholesterol esterase and it will be broken down to cholesterol and fatty acid. Okay, so after all these things are broken down, now what happens? So all these digested forms, that is fatty acids that are formed, monoacyl glycerol, we have said that 75% remains as monoacyl glycerol, cholesterol, and then another very important thing, that is fat soluble vitamins. There are 4 fat soluble vitamins, as you all know, vitamin A, D, E, and K. They are also absorbed along with fat, obviously. So all these things after digestion, they form a structure that is known as micelle or micelle, whatever you pronounce. So you know this is formed, and the most important thing that helps to form this is bile acids. So we all know that. So with help of bile acids, these things, they form a structure known as micelle, and in that form they get absorbed. So the fat absorption depends upon a proper formation of this micelle. In fact, this micelle formation also helps in the digestion also, but we'll not go into that detail. But here one interesting thing is short chain fatty acids and medium chain fatty acids. Short chain and medium chain fatty acids, that is SCFA and MCFA. These things, they are more or less water soluble. So they do not need these micelle formations to get absorbed. So they can get absorbed rather directly. So they do not usually need help of this micelle formation. So in micelle, as you know, it is typically formed by amphipathic lipids of this kind, where a structure like this is formed, where the outside is formed by the hydrophilic or polar head groups of the amphipathic lipids. You know, in the amphipathic lipids, the polar head and then there is non-polar tail. So here comes the main role of bile acids, which is the most important amphipathic structure here or molecule here. So here the hydrophilic polar head groups are towards the outside and hydrophobic non-polar tails, they are towards the inside. So what happens? This outside structure is water soluble, so they can help in their absorption. On the other hand, this inside is hydrophobic, where all these lipid structures, that is fatty acid, MAG, cholesterol, vitamins, etc., all of them, they can safely reside here. So my lipid structures, they are resting here, and with this outside hydrophobic coat like structure, they help in their absorption. If we try to see a 3D like structure of this, then we can see that this is like the 3D structure, where the outside, these are the hydrophilic polar heads of these structures, these are the amphipathic molecules, and this inside this area, these are covered by the hydrophobic non-polar tails. So all my fats that are going to be absorbed, they remain here inside in an environment that is non-polar, where they are comfortably staying. But because the outside is polar, they can get absorbed. Okay, so now this entire absorption process I have drawn and put a schematic diagram, rather simplified schematic diagram. In different books you will find different diagrams. They are obviously a little bit complicated, there are more informations. I have simplified it, so obviously some of the informations I have left, I have not given. I've intentionally left out some of the informations to make it more simplified. So the overall process is obviously more complicated, but this schematic diagram I think will help you to understand, and if you can reproduce this one in exam, that would be good enough for you. Okay, so let us see what we have discussed. I have given that only in this diagram. So see, this part is there are three compartments. This part is the intestinal lumen, this part is the enterocyte cell, and this is from the enterocyte cell. It is absorbed, it can be absorbed into the lymph vessels, it can be absorbed into the bloodstream, that is the portal blood. Okay, so in the intestinal lumen, see from our diet what comes. The main three things: this is the TG, this is the cholesterol ester, this is the fat soluble vitamins. Okay, the phospholipids I have not included in this diagram, but they will also be there. So the TG is broken down into mainly by the pancreatic lipase into monoacyl glycerol and fatty acid. We have discussed that. This monoacyl glycerol, 75% will remain as such, 25% will be broken down to glycerol and fatty acid, about 25%. Okay, fat soluble vitamins, they are not, nothing is happening, they are remaining as such, and cholesterol ester is broken down by cholesterol esterase into cholesterol and fatty acid. Okay, so now what, see, the fatty acid from all these places, from here, from here, from here, all the fatty acids, they are coming, then all the fat soluble vitamins, they are coming, the cholesterol is coming, and monoacyl glycerol is coming. All these molecules, they assemble together with help of the main thing, that is the bile acids. With help of bile acids, all these digested forms, they assemble and form a structure that is known as micelle, that we have just seen. And then in that form, they get absorbed. They get absorbed, they enter into the enterocyte cell, and then again, all of them gets free. So the purpose of micelle is to just get them absorbed from the lumen into the enterocyte cell. In the cell, they again are freed. Now what happens, the interesting thing is, the fatty acid now can again recombine with the monoacyl glycerol to form triacyl glycerol, and with cholesterol to form cholesterol ester, and vitamin A, D, K, etc. So all these things, so they were in these forms. So we break them down, get them absorbed, and again get them back into the form, that is the form for storage. That is not storage here, they will be now again converted to this form for further transport. So cholesterol ester, triacylglycerol, vitamins, all of them now assemble to form another very important structure, that is a lipoprotein structure known as chylomicron. Here they assemble with one very important apolipoprotein known as ApoB48, and in this form, this chylomicron form, they get absorbed into the lymph vessels. These are very large structures, so they cannot go into the portal blood. The gap is not good enough for them, so there is fenestration in the lymph vessels, so they can get into the lymph vessels, and from lymph vessels they directly go into the systemic circulation. But this part I am not going to discuss here, because in the lipoprotein classes, the lipoprotein class that will happen there, this structure and formation, and then further where they go, what happens to them, the chylomicron will be discussed in detail, so I'm not going there. So this is what happens with all these structures. So they break down, they get here, they then form chylomicron and go to the lymph vessels. On the other hand, there are some structures which, or some molecules which are more or less water soluble. So we have seen from monoacyl glycerol about 25% breaks down completely. So there glycerol is formed. So the glycerol itself can get directly absorbed into the enterocyte cells and can directly get absorbed into the portal blood. So here it goes into the bloodstream. And as I have mentioned, small chain and medium chain fatty acids, they also can directly get absorbed into the enterocytes and then into the portal blood. So majority of the lipid we see, they get absorbed into the enterocyte through micelle, and then once inside the enterocyte, they form a structure known as chylomicron, and in that form they get absorbed into the lymph vessel. So majority of our lipids are absorbed into the lymph vessels, and through lymph vessels they directly go to the systemic circulation. So most of the things, most of our diets, nutrients that we eat through our intestine, they usually go to the portal blood and then to liver first. But lipids mostly are an exception, where they get into the lymph vessels and then systemic circulation first, as in the form of chylomicron. The only exception being little amount of glycerol, and then whatever amount of small chain and medium chain fatty acids are there, they can directly get into the portal bloodstream. So I think this much of knowledge about digestion and absorption is enough. And with that, we can finish today's class. And thank you very much.